Thank you for the kind introduction. A bit intimidating to be here among world-class experts, and thank you for the invitation. So this presentation is going to be based on a review article, so I'll take the liberty to be a bit more vague, to cover the sort of general principles rather than the tiny details. So let's begin. I have no disclosures, at least I didn't have until today. So we're going to start with a sort of a grim notion of disability-adjusted life years. So between healthy life and death, there is a lot of stuff happening. So you can have disability, you can have disease, and we know that the main contributor to Daly's disability-adjusted life years is cardiovascular disease. And as you can see, in the 1990s, ischemic heart disease and stroke were the main contributors to Daly's, and they remain to do so 30 years later. So nothing has really changed there. So this is why looking at cardiovascular disease is really important, and that's what we're going to do today. Now here you can see statistics on mortality for coronary heart disease and stroke. So red represents women, blue represents men. The solid blue is coronary heart disease, solid red is coronary heart disease, purple is stroke, pink is stroke, okay? So you can see that the stroke is sort of similar. However, you can see a clear difference in coronary heart disease. Men are, and have been for decades, have been dying more from coronary heart disease. However, women tend to live longer than men, so when you sum it up, the effect is sort of similar. So both suffer similarly from cardiovascular disease, but men tend to die more. However, when a man and a woman gets an infarction, a STEMI, an NSTEMI, you can see from this really nice Norwegian study with a 5-year follow-up that women fare worse, okay? So on the other hand, men tend to die more, but women tend to die more after the infarction. So there are sex differences in many ways here. Now, it doesn't mean that the difference is I don't know, in epigenetics, genetics. It could be that women are diagnosed later, they get to the hospital later. It could be a lot of things. But we see a problem here, and we have to do something about it. So why is this? I talked to my mother about this review article we were writing, and I told her, you know, Mom, we're writing this cool review article on the genetic, molecular, and cellular determinants of sex differences in cardiovascular disease. And she told me, Felix, haven't you been studying? You know, there's this hormone called testosterone and estrogen, and that's why women and men are different. And well, she was sort of right? So if you look at this picture from our review article, you can see the effects of estrogen and testosterone on the main organs related to metabolic health and cardiovascular disease. So E represents estrogen, T, testosterone, and plus means increases, minus means decreases, and in the parentheses, you have the effect of excess testosterone, okay? So quite a lot of men use testosterone recreationally, so it's important to think about that as well. So you can see, for example, for the pancreas, both estrogen and testosterone improve insulin sensitivity, okay, when in adequate quantities. Inflammation, both decrease it. For the liver, the LDL to HDL ratio, which is important for health, they're both decreased by adequate levels of estrogen and testosterone, right? In vascular function, we can see that women seem to be advantaged over men. So estrogen definitely improves vascular health, except for thrombosis. As we know, women tend to be at risk for deep vein thrombosis a bit more, and especially estrogen, taking estrogen supplements, contraceptive, for example, can increase thrombosis risk. But the testosterone effects aren't really that bad. You can see it's 0, minus, there's a plus for RAS activation. And if we look at the heart, again, testosterone isn't that bad, right? If you take it in excess, it can predispose you to left ventricular mass. But, okay, so it's not really conclusive yet. We shouldn't forget that women have these women-specific risk factors, right? Reproductive endocrine disorders, polycystic ovary syndrome. Of course, menopause is a big thing. Many things change after menopause. Women become more similar to men in cardiovascular risk, for example. But, many women are premenopausal, and there are still sex differences, and not every woman gets pregnant, and they still have different risks for cardiovascular disease. So although these women-specific factors exist and they are important, they are under-researched, they don't fully explain the differences either. And one more point from this picture, notice that when the hormones come in adequate quantities, they're beneficial for both men and women. If it was all about hormones, we could just give men estrogen, right? We could just give men estrogen, they'll be healthy, if estrogen was good. If testosterone was good, give women testosterone, okay? It doesn't work that way. It's like we have different blueprints that require different hormones to get the healthy response, right? And so this brings us to the genetics and epigenetics and all that kind of stuff. So it's maybe not mostly hormones, or is it? We're not really sure. We're going to look at genetics next, okay? So here we've summarized the different levels of genetic information that you can look at. We heard an excellent talk about the X chromosome just now, and obviously both men and women have X chromosomes. But in humans, sex is determined by the Y chromosome, right? So the Y chromosome determines sex. And the Y chromosome is very interesting because like 95% of it gets passed on to sons directly without change, unless there is a mutation. But mostly it just gets passed on. And because it doesn't really change, when it does change, we get these two different lineages of Y chromosomes, right? And through that, we can construct this tree, evolutionary tree of Y chromosomes that starts, in Africa and gets all these branches. So we give them names like A, B, C. I'm not sure if B and C are, but A, there's K, L, I, all these different letters. And the further you go from Africa. And there is this interesting haplogroup, we call these haplogroups, the different branches. Haplogroup I, especially haplogroup I1, seems to be associated quite robustly with cardiovascular disease. Okay, so the first estimate published in Lancet, I think it was 50% increase in coronary heart disease. A subsequent study found it in the UK Biobank, found it to be a bit less, about maybe 10%, but it's probably somewhere there. I mean, it is increased. And in the UK Biobank study, they looked at different haplogroups, and the haplogroup I1 was the only one associated with cardiovascular risk, okay? So we don't really know why, but there is something interesting going on there. Now, the X chromosome, unfortunately, the X chromosome has had technical difficulties in getting analyzed in GWAS studies and such, because, you have the inactivation, you have escape from silencing, and it's really hard to analyze it correctly. So people have ended up, discarding it purposefully or accidentally analyzing it incorrectly. So we don't have a lot of good data, and good tools have come in the last few years. So I guess there's going to be a lot of good data unless I've missed some important results. But we can tell from chromosome aneuploidies, from Turner syndrome, from Klinefelter syndrome, X0, XXY, that when you have sort of a different amount of X chromosomes from the average, you tend to be predisposed to cardiovascular disease. So Turner syndrome has a predisposition to poor metabolic health, so does Klinefelter syndrome. Okay. Now, personally, I've been looking a lot at the B and C panels. Now, if you look at the B panel, you have a classical GWAS result Manhattan plot. Okay, so the higher you go, the more significant the SNP is, and you have all the chromosomes with all the SNPs going from on the x-axis from left to right. And you can notice there is no X chromosome there. So the point of this, so women are on top, men are on the bottom. The point is that there can be differences in which SNPs are significant between men and women. Now, this doesn't mean that men and women have different frequencies of these SNPs. It just means that when you have the SNP, if you're a woman, you can be predisposed to something. If you're a man, you won't, or vice versa. So here, I've demonstrated it by putting 5 associations for women, 2 for men, and only 1 is shared. Okay, some could be shared, some could not. And this tells us something. Now, it would be interesting to look at this separately, to do candidate gene studies, to use CRISPR in mouse models and see what they do. Or you could summarize the results by using polygenic risk scores, as is done in panel C. So polygenic risk scores, a recap, they combine the effects from single SNPs into this one risk score, and you can stratify individuals, say, top 2%, and the next 10%, middle 60%, and so on. And so here you have plots where you have the high PRS, let's say top 2.5%, the low PRS, bottom 2.5%, and then you have the population average on the dotted line. And you can see when you have more associations, you get more variation in the genetic risk, right? So we've seen this in hypertension, for example. If we construct polygenic risk scores for hypertension, well, for blood pressure, we see that hypertension incidence, the Cox plots look a bit like this. And this is great because you've heard probably that women have, for example, higher heritability for hypertension, say. But what does heritability mean? It means the proportion of variance explained by, say, genetics. Okay, well, by genetics. But it doesn't mean, it's not an absolute measure. So it could be that men have more variation in environmental risk factors, and even though the genetic component is the same, because men have more environmental variation, let's say, their genetic proportion is going to be smaller than for women. But polygenic risk scores give you a quantitative tool to look at how much is there actually variation. You can even quantify it. So usually they're standardized, but you don't have to standardize them. You can use unstandardized polygenic risk scores if you want to. Okay, so these are the different levels that I thought we should sort of acknowledge. Now let's go into the epigenetic findings, which we summarized in our review article. Most of the data comes from this magnificent systematic review by Hartmann, Huisman, and Reuter. Unfortunately, the results were kind of skim. There were some maybe promising, like men have more epigenetic aging in blood, saliva, and brain tissue. Okay, there was one study showing that. I'm not sure how much you can make of that. One of the problems is that these studies usually reporting sex differences in epigenetics. By the way, it's all DNA methylation. These were all DNA methylation, 13 studies. They're not prospective. They're usually cross-sectional. And when you have a cross-sectional study, there is this issue that when in genetics, okay, cross-sectional might work, genetics doesn't change. When you have a cross-sectional study in epigenetics, you have to remember that the environment also modifies epigenetics. So if you have a disease, it might be that before the disease you had a different epigenetic profile that predisposed you to the disease, and then the disease changed it, for example. That could be the case. So you have to be really careful. There was this nice study showing that DNA methylation in blood leukocyte DNA predicts, so this was a prospective study. I'm sorry if I messed up the terminology. There were cross-sectional studies, the 13 studies, and this is a prospective study, yeah? So we have prediction here, and they show that methylation does predict future myocardial infarction and coronary heart disease. And here they pointed out the problem that many of these studies have. Their findings didn't really agree with previous literature, which has been cross-sectional, right? So they made the comment that they should be prospective, and we did a prospective study, and we showed that there is a predictor of future risk for myocardial infarction. But unfortunately, they did a race-specific analysis, but not a sex-specific analysis. It's great that they did a race-specific analysis, but of course we would like them to have done a sex-specific analysis as well. So they adjusted for sex, and we don't really know of differences here. Now, I want to bring up the discussion from the systematic review on these epigenetic sex differences. When your discussion starts out like this, you didn't really get what you maybe expected. So they say we highlight the insufficient amount of sex stratification performed in cardiovascular epigenetic research. Okay? They go on to say that these studies in cardiovascular epigenetics do not show their data stratified by sex despite the well-known sex differences in CVD, okay? So they're really beating it into that this is not okay. And in the papers describing only one sex, 86% were in men and only 14% in women. So you can see that women are underrepresented here. I really want to, if one thing you could take from this presentation, it's that when you go back to your labs, talk to your colleagues, remind them it doesn't take a lot to do a sex-specific analysis, and if you've done it, it didn't look good, include it as a supplement number 11 if that's what you have to do. But please, we need people to do it so that people have data to do meta-analysis and further downstream analysis on. This is really important. This systematic review found 75 studies having the adequate data but not doing sex stratification, and they ended up with 13 studies doing sex stratification. Okay, so there was a lot more potential that wasn't realized. So this is not okay, right? And we have to do something about it. And we really can by including the data. Now, this is the famous Inkblot test where they test what you see, and the psychologists ask you, what do you see? I tend to see risk scores everywhere because I like to do polygenic risk scores. So I see polygenic risk scores in methylation data as well. And let's not forget this symposium is about going from the molecule to bedside. And I think one way of doing it could be prediction. So as part of prevention, how do you go from prediction to prevention if you have a polygenic risk score? You always hear, oh, that's great, you have a polygenic risk score, what are you going to do with it? A clinician asks you, how am I going to use it? And for a long time, I've given answers like, well, maybe we could do this and maybe we could do that, but they're never convinced. Now we have a study from Finland showing that communicating polygenic and clinical risk for atherosclerosis actually has an impact on health behavior. So what they did was they had these people come into the clinic and they assessed their cardiovascular risk through the AHA risk equation and these polygenic risk scores. And they explained both to the patient, okay? And then the patient went home and they went on with their life. And then they asked them to come back and asked, well, did you make any health changes? And the people at high risk, at high combined risk for polygenic risk and clinical risk, they found that 42.6% had made some significant change. For example, there could be, lose weight, stop smoking, stuff like that. Some hired a personal coach for themselves. In the average and low risk groups, only 33% had made a change. So there was a change of almost 10 percentage units. And they saw that from the effect, genomic risk took around 1.10 odds ratios out of it. So the polygenic risk had a contribution to this. It's probably less than the clinical risk score, as is always with genetics and clinical data, but there is an effect, and this is something that we shouldn't ignore, because on population health level, as we heard in the previous speech, public health is in science where small effects can have large consequences, right? Now, I want to just emphasize, I know you probably know this already, but emphasize the parallels between genetics and epigenetics. We already have really great pipelines for genetics. A lot of expertise, a lot of pipelines, a lot of software that can be pretty easily converted to epigenetics. And why do I say this? Well, when in genetics you have these SNPs, right? In epigenetics you have the methylation sites. Talking about methylation now because that's the most common one. So CpG site methylation, so it could be no methylation, some methylation, high methylation. And for SNPs, you have zero of the effect variant, one effect variant, two effect variants, so very similar there. You can run association studies, you can run a GWAS for genetics, you can run an genome-wide epigenetic analysis for methylation. And then you can do a PRS, polygenic risk score, or you can do an MRS, methylation risk score. And I know these have been done, but just emphasize that these are very similar. However, there are differences, right? When you try to do genetic studies, you can take the DNA from anywhere. You can take saliva, you can take blood, you can take anything. It's going to be the same basically, with some exceptions, obviously, but pretty much always the same. In epigenetics, it matters where you take the data out, right? Blood is different from saliva, it's different from brain tissue, as we just mentioned before. Also, age has a huge impact, sex has an impact, lifestyle has an impact, many things have an impact. So if you're going to start doing association analyses to investigate which methylation sites have an association with a disease, you really, if you want to be technical about it, you have to do all these different analyses with different cohorts, and you're going to get more accurate results the more similar the cohorts are to the population you're studying. But there is another advantage to epigenetics. Well, there is an advantage to epigenetics that genetics doesn't have. So we have Mendelian randomization. Mendelian randomization is this concept where you use genetics and SNPs to sort of identify these naturally occurring randomized trials to make deductions like, okay, actually HDL isn't causally related to cardiovascular disease, but LDL is. So that's one of the things we've learned recently. And drug companies obviously love this because all the pharmaceutical research that has gone into HDL drugs is pretty much, well, I'm not going to say useless, but of questionable use if it's not causally linked to cardiovascular disease, the endpoint that we're looking at. So the idea is that for methylation, well, for epigenetics, you could do this kind of a two-step process where first you establish the causal connection between an exposure and a CpG site. And then in this next step, you're going to establish the causality between the CpG site and the phenotype, say cardiovascular disease, hypertension. And when you combine these, you, by mathematical logic, get that there should be a causal connection between the exposure and the phenotype. But also you just, then you know that, okay, this exposure changes the epigenetics, and if the epigenetics is changed and it has a causal connection to the phenotype, then by changing the methylation, you should be able to also affect the phenotype. So you know better than I what kind of tools we could have to edit methylation or edit epigenetics in the future. But this is something we can't really have with genetics. In genetics, we have an issue with GWAS results. We never really know, is this causal or not? Is this SNP causal? We can do, we can do fine mapping. It can give us some idea. But now we can also, with CRISPR, we can do gene editing in mice, as we talked yesterday with a colleague, that you can really nowadays investigate it directly, and actually it was mentioned today. So new things are happening all the time. But I think it's exciting that we can establish causality with relative ease, because Mendelian randomization can be done online with a tool like MRBase. It's a website where you just input your preferences and it does the first analysis for you, and then you can go home and do the actual paper. Now I want to acknowledge my colleagues who were writing this paper with me, and Munira and my supervisor unfortunately couldn't be here today, but I hope I could give you at least some general overview, food for thought. And for some reason, this didn't have the acknowledgements to the organizers. I want to thank the organizers for this great event. I mean, it's just amazing, and looking forward to the symposium today, tomorrow, and the day after tomorrow. Thank you.